Hi everyone! Today we're diving into Chapter 19, Biochemistry. I know the word biochemistry might sound intimidating, but don't worry. We're going to break it down in a way that's clear, relevant, and connected to nursing. Biochemistry is the study of the chemical processes in living organisms. That means everything from how your body digests food, to how your brain sends signals, to how medications work in the bloodstream. It's all biochemistry. So let's get started and explore how the chemistry of life connects directly to patient care. Let's begin by understanding the big picture, the Human Genome Project. This was a massive scientific effort that helped us understand what makes us who we are at the genetic level. Imagine your body as a house and your DNA as the blueprint. That blueprint is passed from parents to children through genes. Every cell in your body has this instruction manual, and those instructions control everything from your eye color to your risk for certain diseases. NCLEX tip: genetic predispositions often impact disease risk. For example, some patients may be genetically more likely to develop certain cancers or metabolic conditions. This project took 15 years and helped us map out every gene in the human body, about 20,000 to 25,000 in total. Why does that matter for nursing? Because it explains why some patients respond differently to drugs. It also helps us predict who might be more likely to get diseases like breast cancer or Alzheimer's based on small differences in their genes, called single nucleotide polymorphisms or SNPs. NCLEX practice question. Which of the following best describes the goal of the Human Genome Project? A. To find a cure for all genetic diseases. B. To map the entire DNA sequence of the human genome. C. To create synthetic genes. D. To eliminate genetic disorders. Correct answer, B. Explanation: the goal was to map the human genome, not to cure diseases directly, but to provide a roadmap for understanding them. Now here's a real nursing connection. Certain drugs are made using human genes. Take interferon, a drug used for multiple sclerosis. Scientists took the gene that makes interferon, inserted it into bacteria, and now those bacteria produce the drug. It's a little like baking cookies using someone else's recipe. The recipe is the gene and the bacteria are the bakers. This kind of drug production is called recombinant DNA technology. In clinical practice, this is the foundation for biologics like insulin, vaccines, and some cancer treatments. Let's zoom into the basic unit of life, the cell. You can think of a cell like a tiny hospital. The nucleus is the administration center. It stores all the instructions, DNA. The cell membrane is like the hospital walls. It keeps things in and out. The cytoplasm is the working area where the real action happens, with different departments or organelles doing specific jobs. Understanding the cell helps us understand disease, medication delivery, and more. NCLEX connection. Many medications work by targeting specific cell structures. For example, chemotherapy targets rapidly dividing cells by interfering with the nucleus and DNA. Let's take another look at the cell. Remember, it's the smallest unit of life. If the body is a hospital, then a cell is like a single room with its own little systems running inside. Some cells have a nucleus, which is like the control center that stores the instructions. The outer wall of the cell is the cell membrane. It holds everything together and decides what comes in and what goes out. Between the nucleus and the membrane is the cytoplasm, like the room space where workers, called organelles, do their jobs. These parts work together to keep the cell and ultimately the patient alive and functioning. Cells are made of 4 major types of chemicals: carbohydrates, lipids, proteins, and nucleic acids. These are like the building blocks and energy sources for everything the body does. Think of them as the essential ingredients in a recipe for life. As a nurse, when we talk about nutrition, energy levels, cell repair, or even medications, these are the molecules we're really dealing with. Let's start with carbohydrates. These are the body's main short-term energy source. If you haven't eaten in a while and suddenly have a piece of bread or fruit, you'll feel more alert because your body is using that carbohydrate for quick energy. Carbs are also structural. Plants use them to build their bodies, like the fiber in vegetables. Carbohydrates usually follow a basic formula, kind of like repeating units of sugar, and chemically they're based on structures called aldehydes or ketones with a lot of OH groups, which help make them soluble in water. Now let's get specific. Glucose is a type of sugar that your body uses as its main fuel. Think of it as gasoline for your cells. It's an aldehyde sugar, meaning it has a particular group of atoms, CHO, at the end. It also has a bunch of OH groups, which makes it dissolve easily in water and blood. That's great because it needs to travel through your bloodstream to reach all your cells. NCLEX style question? Let me ask you this. If a patient is NPO, nothing by mouth, for 12 hours, what molecule will their cells primarily use for energy once all food glucose is used up? A. Protein. B. Fat. C. Glucose stored as glycogen. D. DNA. The correct answer is C. Glucose stored as glycogen. The liver stores excess glucose in the form of glycogen, which can be broken down when needed for energy. Glucose is what we call a monosaccharide. That means it's a single sugar unit and it can't be broken down into smaller sugars. Think of it like a Lego block. You can use one on its own, or you can snap it together with others to build bigger structures. In water, glucose doesn't just float around in a straight line, it rearranges into a ring shape, which is how it exists in the bloodstream and in your cells. Let's look at some more sugars. Glucose, fructose, and galactose all have the same formula, C6H12O6, but they're arranged slightly differently. We call these sugars isomers, same ingredients, different shapes. These are all hexaces, meaning they each have 6 carbon atoms. These slight changes in shape may seem tiny, but in biochemistry, shape matters a lot. For example, even though fructose and glucose both give us energy, our bodies process them a little differently. It's like using different keys for different locks. So what's special about each one? Fructose is the sugar naturally found in fruits and honey. That's why fruits taste sweet. It's fructose. Galactose is found in milk and also in brain tissues, so it's sometimes called brain sugar. Glucose is the primary sugar our cells love and depend on. NCLEX style question? Let me quiz you. A nurse is reviewing the chart of a patient with galactosemia, a condition where the body cannot process galactose. What dietary adjustment is most appropriate? A. Increase fiber intake. B. Eliminate dairy products. C. Reduce protein consumption. D. Avoid red meat. The correct answer is B. Eliminate dairy products. Galactose is found in milk, sugar, or lactose, so avoiding dairy is essential. Now that we know about single sugars, let's see how they come together. When 2 monosaccharides like glucose and fructose combine, they form a disaccharide, a 2-sugar unit. But here's the key part. When they combine, a water molecule is removed in a process called condensation, and they link through something called a glycosidic bond. In digestion, your body breaks that bond to get the individual sugars back, so they can pass through the intestinal wall and enter the bloodstream. This is why enzymes like lactase are so important. They help break these bonds in the gut. Let's look at a real-life example, sucrose or table sugar. It's made by linking 1 glucose and 1 fructose molecule together. That's the sugar you put in your coffee or find in candy. Once in your body, enzymes break the bond so glucose and fructose can be used for energy. Clinical relevance. In diabetic patients, keeping track of how much sucrose they consume is important because it quickly raises blood sugar levels. Okay, now we go big. When many monosaccharides are linked together, we get polysaccharides. These are long chains of sugar units, and they're a type of polymer. Just like a pearl necklace is made of many beads, a polysaccharide is made of many sugar units. These include things like starch, cellulose, and glycogen. Why does this matter for nursing? Because understanding these molecules helps you understand how the body stores energy or why fiber is so important for digestion. NCLEX style question? What is the function of glycogen in the human body? A. Long-term storage of genetic material. B. Storage form of fat. C. Storage form of protein. D. Storage form of glucose. The correct answer is D. Storage form of glucose. Glycogen is how your liver and muscles store excess glucose for later energy use. Let's organize all these carbs we've been talking about. Monosaccharides and disaccharides like glucose and sucrose are considered simple carbohydrates. They provide quick bursts of energy. Polysaccharides like starch, cellulose, and glycogen are complex carbohydrates. They release energy more slowly and can also serve structural roles. Starch and cellulose are made of repeating glucose units, but they behave very differently, which we'll see in the next slide. Now this is interesting. Starch and cellulose are both made entirely of glucose, but the way the glucose units are linked makes all the difference. In starch, the links are called alpha linkages, and they're digestible by humans. In cellulose, the links are beta linkages, and we can't digest those. Think of starch as soft and chewable, like potatoes and rice, and cellulose as tough and fibrous, like celery and leafy greens. Even though we can't digest cellulose, it's still important as fiber, helping to keep your digestive system healthy. Here's the practical difference. Starch is digestible. Your body breaks it down into glucose to use for energy. Cellulose, however, passes through your gut undigested. That's why fiber-rich foods help prevent constipation and promote gut health. NCLEX style question. A patient is prescribed a high-fiber diet. Which of the following best explains the benefit of cellulose in this diet? A. It provides essential amino acids. B. It's a fast source of glucose. C. It enhances water absorption and bowel movement. D. It raises blood pressure. The correct answer is C. Cellulose helps draw water into the intestines and adds bulk to stool, promoting bowel regularity. Now let's move on to another class of biomolecules, lipids. These include fats, oils, and even steroids. What makes lipids special is that they don't mix with water. They're insoluble in water but dissolve in things like oils and alcohols. This is actually really useful in the body. Lipids are great for long-term energy storage, insulation, and especially for building cell membranes, those outer barriers we talked about in the cell slides. One type of lipid is the fatty acid. These are long molecules with a carboxylic acid group on one end and a hydrocarbon tail on the other. That hydrocarbon tail is like a long string of carbon atoms, and it's the reason fatty acids don't mix with water. We'll soon look at how these fatty acids form larger structures. But for now, just remember, fatty acids are the basic units of many lipids in your body. Now we get into the types of fatty acids. Fatty acids come in two main forms. Saturated fatty acids, like myristic acid, have no double bonds in their carbon chains. That means all the carbon atoms are saturated with hydrogen. They tend to be solid at room temperature, like butter or lard. Unsaturated fatty acids, like oleic acid, have one or more double bonds. This creates bends or kinks in the chain, which makes them more fluid and usually liquid at room temperature. Think olive oil. The type of fat matters for heart health. Too much saturated fat. That increases the risk of atherosclerosis, clogged arteries. To visualize it, saturated fats are like straight straws that stack tightly together, solid and dense. Unsaturated fats are like bent straws that don't pack tightly, more fluid. That's why animal fats like beef fat are solid, while plant oils like olive oil are liquid. From a nursing standpoint, patient diets rich in unsaturated fats are generally healthier. NCLEX style question. Which of the following dietary fats is considered healthiest for a patient at risk for coronary artery disease? A. Butter. B. Lard. C. Olive oil. D. Bacon fat. The correct answer is C. Olive oil. It contains monounsaturated fatty acids, which are associated with improved heart health. Here's a chemical twist. Fatty acids all have the same base structure, but they differ in the length and shape of their hydrocarbon tails, or R groups. These differences determine their melting points, how they're stored, and how the body uses them. Remember, the longer and straighter the tail, the more solid the fat tends to be. Here's a chemical twist. Fatty acids all have the same base structure, but they differ in the length and shape of their hydrocarbon tails, or R groups. These differences determine their melting points, how they're stored, and how the body uses them. Remember, the longer and straighter the tail, the more solid the fat tends to be. Now let's connect fatty acids to something familiar, fats and oils. These are called triglycerides. They're made of glycerol, a 3-carbon molecule connected to 3 fatty acids. That's why we call them triglycerides. You can think of it like a backbone, glycerol holding 3 tails, fatty acids. Whether it's solid fat or liquid oil depends on whether those tails are saturated or unsaturated. So how are triglycerides formed? Through a chemical reaction between glycerol and 3 fatty acids. This reaction removes water, a condensation reaction, and forms ester bonds which hold the structure together. This is the basic chemical makeup of the fats and oils we eat, and also the fats stored in our body. Clinical connection. When a patient has high triglyceride levels in a lipid panel, it means their body has too much fat stored or circulating, which is a risk factor for metabolic syndrome and cardiovascular disease. Here's a real-life example of a triglyceride, tristerin, which is found in beef fat. It's made from glycerol and 3 stearic acid molecules. This is a classic example of a saturated fat, meaning all the fatty acid tails are straight and tightly packed. That's why beef fat is solid at room temperature. As a nurse, you'll see how dietary fats like this can impact cholesterol levels and cardiovascular health. To summarize, if the fatty acids are saturated, the triglyceride is usually a solid fat. Think lard, butter, animal fat. If the fatty acids are unsaturated, the triglyceride is a liquid oil. Think canola oil, olive oil, or sunflower oil. This is important in nutrition and patient education. We often advise patients to reduce saturated fat intake and switch to healthy unsaturated fats to lower their LDL cholesterol and reduce heart disease risk. Phospholipids are another important kind of lipid. Structurally, they're similar to triglycerides, but one of the 3 fatty acid tails is replaced with a phosphate group. This phosphate group is polar, meaning it mixes with water, while the rest of the molecule is nonpolar, it repels water. So a phospholipid has a hydrophilic head and hydrophobic tails. That's what allows them to form cell membranes, creating a barrier that holds the cell together. Let's zoom in on that. A phosphatidylcholine, a common phospholipid in cell membranes, has a polar head that loves water, hydrophilic, 2 nonpolar tails that avoid water, hydrophobic. When phospholipids line up, the heads face outward toward the watery environment and the tails hide inside. This is the basis of the lipid bilayer in all of our cells. There's another similar group called glycolipids. These also have a fatty tail and a polar head, but instead of a phosphate group, they have a sugar molecule. Both phospholipids and glycolipids are perfect for forming cell membranes because they create a waterproof barrier around the cell. Their dual nature, part that loves water and part that fears it, is key to building that protective shell. NCLEX style question? Which feature of phospholipids makes them ideal for forming cell membranes? A. Their ability to conduct electricity. B. Their dual polarity, hydrophilic heads and hydrophobic tails. C. Their ability to bond with proteins. D. Their lack of chemical reactivity. The correct answer is B. The hydrophilic head interacts with water while the hydrophobic tails avoid it. This dual nature allows phospholipids to form a bilayer structure that protects and encloses cells. Let's put it all together. The cell membrane is made of a lipid bilayer, which means 2 layers of phospholipids or glycolipids. The hydrophilic, water-loving heads face the outside and inside of the cell where the water is. The hydrophobic, water-fearing tails point inward, away from water. This structure keeps the cell's contents safe and allows selective movement of substances in and out, like oxygen in and waste out. It also plays a critical role in medication delivery because some drugs have to cross this barrier to be effective. Another class of lipids includes steroids. They look very different. They have a 4-ring structure. The most well-known steroid is cholesterol, but others include testosterone and estrogen, which are hormones. These molecules have powerful effects in the body. Even tiny amounts can regulate growth, reproduction, and metabolism. Let's talk about cholesterol. It helps stabilize cell membranes. It's also a precursor for making other important hormones like testosterone and estrogen. That's why cholesterol is essential for life, but too much cholesterol in the blood, especially LDL or bad cholesterol, increases the risk for atherosclerosis and heart attacks. NCLEX style question? Which of the following best describes the role of cholesterol in the body? A. It stores genetic material. B. It provides short-term energy. C. It's part of cell membranes and a precursor to hormones. D. It transports oxygen in the blood. The correct answer is C. Cholesterol is a structural component of membranes and is used to synthesize important steroid hormones. This slide connects everything to patient care. Most fats and oils in our diet are triglycerides. During digestion, they're broken down into fatty acids and glycerol, then rebuilt and absorbed. Because this process is slower than digesting carbs or proteins, fat gives a longer-lasting feeling of fullness. The FDA recommends that fats make up less than 30% of total calories and saturated fats should be less than 10%. Why? Because high saturated fat intake is linked to heart disease. Meanwhile, monounsaturated fats like olive oil may actually be protective. Now we're moving on to a new type of biomolecule, proteins. And trust me, proteins are everywhere. They build your muscles, skin, and cartilage. They're your enzymes, your hormones, your antibodies, and even the oxygen carriers in your blood, like hemoglobin. Proteins basically do all the work that keeps your body functioning. That's why understanding their structure and function is essential in nursing. So what exactly are proteins? Proteins are polymers. That means they're long chains, made up of amino acids, which are like building blocks. Each amino acid has 3 parts: an amine group, a carboxylic acid group, and an R group, also called a side chain, which is what makes each amino acid unique. It's like a necklace made of beads. Each bead is an amino acid, and the R group is what gives each bead a different shape and function. Now let's look closer at these R groups or side chains. They can be nonpolar, like in alanine, think water repelling; polar, like in serine, water attracting; acidic, like aspartic acid; or basic, like lysine. Why does this matter? Because the type of R groups influences how the protein folds and what job it can do in the body. There are 20 standard amino acids that your body uses to build all kinds of proteins. Some are essential, meaning you have to get them from food, like in meat, eggs, or beans. Others your body can make on its own. Every protein in your body, from enzymes to antibodies, is built from some combination of these 20 amino acids. How do we go from amino acids to proteins? Amino acids link up when the amine group of one bonds with the carboxylic acid group of another. This bond is called a peptide bond. 2 amino acids linked together is a dipeptide. Many linked together is a polypeptide. A complete functional structure is a protein, which may contain hundreds or even thousands of amino acids. Now comes something super important. Protein shape matters. Why? Because if a protein doesn't fold correctly, it can't do its job. For example, insulin must fit exactly into insulin receptors on cells. If insulin had a different shape, it wouldn't work and blood sugar would stay dangerously high. Protein folding is influenced by the amino acid side chains, and it determines whether a protein becomes a muscle builder, an enzyme, or an immune defender. Proteins don't just stop at a simple chain of amino acids. They fold and twist into complicated shapes. We analyze protein structure at 4 levels. Primary structure, the straight chain of amino acids. Secondary structure, small patterns like spirals, alpha helix, and pleats, beta sheet. Tertiary structure, the whole 3D folded shape of one chain. Quaternary structure, how multiple protein chains come together to form one functional unit. Each level plays a role in how the protein behaves in the body. Let's look closely at primary structure. This is the exact sequence of amino acids in a protein chain. It's like letters in a sentence. If you change even one, the meaning could be totally different. For example, part of the insulin protein might look like this: glyylvalgluclancies. If you accidentally swap just one amino acid, it could ruin the entire function of the protein. And yes, your body reads this exact order from your DNA. Here's why that sequence matters. In sickle cell anemia, just one amino acid in hemoglobin is changed, from glutamic acid to valine. That one change causes red blood cells to sickle or bend into a crescent shape. These misshapen cells can get stuck in blood vessels and lead to pain, organ damage, and anemia. So even a single mistake in the primary structure can lead to a serious genetic disease. Moving up a level, now we talk about secondary structure. One common shape is the alpha helix, a spiral coil. This structure is held together by hydrogen bonds between nearby parts of the protein backbone. Think of it like a slinky or a curled ribbon, very stable and stretchy. Keratin, the protein in your hair and nails, is mostly made of alpha helices. That's why they're tough yet flexible. Another secondary structure is the beta pleated sheet. Instead of coiling, this pattern lies flat and zigzags like an accordion. It's also stabilized by hydrogen bonds, but this time between strands lying side by side. Silk, for example, is made of proteins with beta-pleated sheet structures. That's what gives it strength and smoothness. NCLEX style question? A nurse is teaching a patient about sickle cell disease. Which of the following best explains the cause of the condition at the molecular level? A. Protein chains are too short. B. There is an imbalance in calcium ions. C. One amino acid in the hemoglobin protein is substituted. D. The protein is missing all hydrogen bonds. The correct answer is C. A single amino acid substitution in hemoglobin causes the red blood cells to change shape and malfunction. Now, most proteins don't stick to just one pattern. Some areas might be alpha helices, others might be beta sheets, and some may not have a regular pattern at all. These are called random coils. For example, keratin, like in hair, is mostly alpha helix. Silk is mostly beta sheet. Other proteins have a mix. These folding patterns help determine how the protein functions, whether it's flexible, firm, stretchy, or strong. Here's a fun and relatable application: hair. Your hair is made of keratin, which is packed in a coiled alpha helix structure, stabilized by hydrogen bonds. When hair gets wet, water interferes with those hydrogen bonds. The structure relaxes and the hair stretches out. It actually becomes about 10 to 12% longer when fully wet. This is why your ponytail feels looser after a shower. It's a great real-world example of how protein structure can change depending on the environment. Now let's go deeper. The tertiary structure is the protein's complete 3D shape, resulting from interactions between R groups that are far apart in the chain. These interactions include hydrogen bonds between polar groups, disulfide linkages, covalent bonds between sulfur atoms and cysteine, hydrophobic interactions, nonpolar side chains grouped together, salt bridges between acidic and basic R groups. This 3D shape is what allows proteins to bind to receptors, enzymes to catalyze reactions, and antibodies to recognize invaders. Tertiary structure also determines whether a protein is a fibrous protein like keratin or collagen, which is water insoluble and used for structural support, or a globular protein like hemoglobin or enzymes, which are water soluble and travel through your blood. Understanding this difference helps explain why some proteins stay put, like in skin or tendons, and others move around to do their jobs, like oxygen transport or immune defense. Finally, some proteins are made up of multiple chains called subunits. When these subunits come together, we describe the arrangement as quaternary structure. A great example is hemoglobin, which has 4 subunits. The way they fit together determines whether hemoglobin can pick up and release oxygen efficiently. And just like in tertiary structure, quaternary structure is stabilized by hydrogen bonds, disulfide bonds, hydrophobic interactions, and salt bridges. NCLEX style question. A nurse is explaining protein structure to a student. Which level of structure is affected when multiple protein chains interact to form a functional protein like hemoglobin? A. Primary. B. Secondary. C. Tertiary. D. Quaternary. The correct answer is D. Quaternary structure refers to how multiple polypeptide chains are arranged in a multi-subunit protein like hemoglobin. Let's pause and review what keeps these complicated protein shapes stable, especially tertiary and quaternary structures. The interactions include hydrogen bonds between polar side chains, disulfide bonds, strong covalent bonds between sulfur atoms, hydrophobic interactions, nonpolar R groups hiding from water, salt bridges, ionic bonds between acidic and basic side chains. These forces work like scaffolding, giving proteins the exact 3D shape needed to do their job. Here's a quick summary of all 4 protein structures. Primary, the specific sequence of amino acids. It's like spelling a word. Secondary, regular shapes like coils, alpha helix, or sheets, beta pleated sheet, formed by nearby amino acids. Tertiary, the complete 3D shape of a single chain, influenced by long-distance interactions between side chains. Quaternary, how multiple chains come together to form one functional protein. Each level of structure is crucial for the protein to function properly. A small error in any one level can lead to disease. Now we move on to nucleic acids, the body's blueprint writers. They tell the body what proteins to make and when. There are 2 main types. DNA, found mostly in the nucleus, it stores the genetic code. RNA, found throughout the cell. It helps carry out the instructions from DNA. Just like proteins are made of amino acids, nucleic acids are made of building blocks called nucleotides. Each nucleotide has three parts: a phosphate group, a sugar either ribose in RNA or deoxyribose in DNA, a nitrogen-containing base. These nucleotides are like letters in an alphabet. When linked together, they spell out the instructions for making proteins. DNA is a polymer of nucleotides. Every DNA nucleotide includes a sugar, deoxyribose, a phosphate group, one of four bases: adenine A, cytosine C, guanine G, or thymine T. These nucleotides are linked through phosphate bonds, creating a long, stable chain. The sequence of these bases is what forms genes, which act like recipes for making specific proteins. NCLEX-style question. A nurse is reviewing cell biology with a student. Which component of a DNA nucleotide determines the genetic code? A, the sugar molecule. B, the phosphate group. C, the nitrogen-containing base. D, the peptide bond. The correct answer is C, the nitrogen-containing base. A, T, C, or G determines the genetic code. The sequence of these bases forms genes. Let's focus on the bases in DNA. There are four: adenine A, thymine T, cytosine C, guanine G. Each base pairs with only one specific partner. A pairs with T, C pairs with G. These pairings are like magnets. They're held together by hydrogen bonds and are what make DNA such a stable double helix. So how do these bases work? Each three-base sequence is called a codon, and it codes for one amino acid in a protein. For example, the codon AGT codes for the amino acid serine. TGA codes for threonine. There are only four bases, but using three-letter combos gives 64 possible codons, more than enough to code for all 20 amino acids. This coding system is called the genetic code. It's like a universal translator. The same codons mean the same amino acids in humans, rats, or even bacteria. This is exactly why we can insert human genes into bacteria and get them to produce insulin or other proteins, because the genetic code is shared. A gene is just a long sequence of codons that tells the cell how to make a specific protein. Think of it like a sentence. Each codon is a three-letter word. Together they form instructions for making a protein. A single gene might have hundreds or thousands of codons, depending on the size of the protein it codes for. Genes are stored in structures called chromosomes, and we humans have 46 chromosomes. Let's map this out from smallest to largest. Nucleotide, the smallest building block. Codon, a three-nucleotide sequence. Gene, a string of codons that code for one protein. Chromosome, a long strand of DNA containing many genes. This hierarchy is key to understanding genetic diseases and therapies like gene editing or genetic testing. NCLEX-style question. A patient asks the nurse what a gene is. What is the most accurate response? A, a chemical that provides short-term energy. B, a three-base sequence that stores fat. C, a section of DNA that contains the instructions to build a specific protein. D, a protein that helps break down nutrients. The correct answer is C. A gene is a section of DNA made up of codons that instruct the body on how to make a specific protein. Let's look at the famous double helix shape of DNA. Imagine a twisted ladder. The sugar and phosphate groups form the sides of the ladder. The nitrogenous bases, A, T, C, G, form the rungs. These bases pair up. A always pairs with T, C always pairs with G, and they connect through hydrogen bonds that hold the two strands together like glue. This specific pairing is what makes DNA able to copy itself perfectly, a process called replication. What's really amazing is that each base can only pair with one other base. This is called complementary base pairing. A with T, C with G. Because of this, when DNA splits during replication, each strand serves as a template to create a new complementary strand. This precision ensures that the new cells after division have exactly the same genetic material. Here's how DNA replication works. The DNA unwinds like unzipping a zipper. Hydrogen bonds between the base pairs break. Each original strand acts as a template. New nucleotides pair with the exposed bases to form two identical DNA molecules. This happens during cell division so that each new cell gets a complete copy of DNA. Think of it like photocopying the instruction manual before handing it to a new worker. Now that we've copied the DNA, let's talk about how proteins are made. This is called protein synthesis. Your body uses DNA instructions to build the proteins it needs. Here's the sequence. You eat protein and it gets broken into amino acids. Your body then uses nucleic acids, DNA and RNA, to rebuild the exact proteins you need. This all happens inside your cells and is directed by your genes. When a cell needs a protein, it doesn't use the DNA directly. Instead, the DNA unzips and makes a temporary copy of the gene as messenger RNA, mRNA. This mRNA leaves the nucleus and travels to a ribosome in the cytoplasm. At the ribosome, the mRNA is read 3 bases at a time. Each codon tells the ribosome which amino acid to add next. As the ribosome moves along the mRNA, it builds the protein amino acid by amino acid. So the whole process is DNA, mRNA, protein. NCLEX style question. Which of the following best describes the role of messenger RNA, mRNA in protein synthesis? A. It carries amino acids to the nucleus. B. It stores energy for protein production. C. It delivers the genetic code from DNA to the ribosome. D. It breaks down dietary protein into amino acids. The correct answer is C. mRNA delivers the genetic instructions from DNA in the nucleus to the ribosome where proteins are assembled. Let's zoom in on what happens at the ribosome during protein synthesis. The mRNA strand moves through the ribosome like a conveyor belt. Each codon, 3 bases, tells the ribosome which amino acid to add. As the ribosome reads the codons, amino acids are brought in and joined together with peptide bonds. This continues until the protein is fully built. It's like following a recipe one step at a time. The final product is a functional protein ready to do its job in the body. Let's tie it all together. DNA holds the master instructions. It passes a copy to mRNA, which exits the nucleus. The mRNA is read by a ribosome, which assembles the amino acids in the correct order. That's how your cells make the exact proteins they need. This process explains how the body builds enzymes, hormones, antibodies, and more. It's also why mutations, small changes in DNA, can lead to faulty proteins and disease. Let's connect this to something familiar, insulin. People with diabetes don't make enough insulin, which helps move glucose from the blood into cells. Without it, blood sugar levels stay dangerously high. For years, we used animal insulin from pigs or cows, but some patients reacted badly. Today, we use human insulin made by bacteria that have been genetically modified to contain the human insulin gene. This is a perfect example of how understanding DNA and protein synthesis leads to better treatments. Here's how we make insulin today. Scientists remove the insulin gene from human cells. They insert it into bacteria. The bacteria copy the gene and produce insulin using their own protein-making machinery. The insulin is harvested, purified, and bottled for diabetic patients. This is called recombinant DNA technology, and it's just one example of how biochemistry saves lives. Let's recap the major players. Carbohydrates give us quick energy and come in forms like glucose and starch. Lipids store energy long-term, build membranes, and include fats, oils, and steroids. Proteins are workhorses, building muscles, acting as enzymes and hormones, and protecting us with antibodies. Nucleic acids, DNA and RNA, store and deliver the genetic code to make all those proteins. It all connects. You eat nutrients, break them down, rebuild what your body needs, carry out life processes. NCLEX style question. Which of the following best explains how human insulin is produced for diabetic patients today? A. It is extracted from cow pancreas. B. It is made by genetically modified bacteria using the human insulin gene. C. It is synthesized by the liver after a high-carb meal. D. It is created when the pancreas receives artificial glucose. The correct answer is B. Today, insulin is produced by bacteria that have been engineered to carry the human insulin gene. This is a product of recombinant DNA technology. Let's quickly reinforce what we learned about protein structure. Primary structure is the exact sequence of amino acids, like spelling words in a sentence. Secondary structure includes patterns like alpha helices and beta sheets formed by hydrogen bonding. Tertiary structure is the overall 3D shape of one protein chain based on side chain interactions. Quaternary structure is how multiple chains come together to make a complete functional protein, like hemoglobin with its 4 subunits. Each level of structure is essential. Change one piece and the protein might not work at all. Now let's summarize how nucleic acids, DNA replication, and protein synthesis work. DNA and RNA are built from nucleotides. DNA contains 4 bases, A, T, C, and G. The order of these bases makes up the genetic code. A codon is a group of 3 bases that codes for 1 amino acid. A gene is a sequence of codons that tells the cell how to build a protein. Genes are stored in chromosomes. We have 46 in total. DNA replication copies the entire genome before cell division. In protein synthesis, DNA is copied to mRNA, which is read at the ribosome and used to assemble the protein. This process is essential to every cell in the human body, and it's the basis of both health and disease. Before we wrap up, let's check in on our goals for this chapter. By now, you should be able to identify the 4 key chemical components of the cell: carbohydrates, lipids, proteins, and nucleic acids. Distinguish between monosaccharides, disaccharides, and polysaccharides. Describe the differences between saturated and unsaturated fats. Recognize how proteins are built from amino acids and describe the 4 levels of protein structure. Understand how DNA and RNA direct the synthesis of proteins. Explain how drugs like insulin are now produced using biotechnology. These are not just science facts, they're clinically important. As nurses, understanding biochemistry helps you interpret lab values, educate patients, and understand how medications and diseases work at the molecular level. Final NCLEX-style question for review. Which of the following best summarizes the flow of genetic information in the body? A. Protein, RNA, DNA. B. DNA, protein, RNA. C. DNA, RNA, protein. D. RNA, DNA, protein. The correct answer is C. The correct sequence is DNA, RNA, protein. DNA holds the code, RNA carries it, and proteins are the result.